So let me just very briefly introduce our speaker today, Dr. Bradley Gordon. He received his Ph.D. in applied physiology from the University of South Carolina, and then did postdoctoral work in cell and molecular physiology at the Penn State College of Medicine. His research is primarily focused on how stimuli such as nutrients, hormones, and physical activity regulate changes in skeletal muscle mass and muscle function in both diseased and non-diseased conditions. And he utilizes both in vivo and in vitro models, including techniques such as synergistic ablation, acute and long-term aerobic exercise training, high-frequency muscle contractions, and obviously not with humans, castration, I hope, to test his hypotheses. So without further ado, let me turn it over to Dr. Gordon, and he's going to be presenting. Oops, hold on, let me let in one other person here.
Thank you, Neil. I appreciate it. I appreciate the opportunity to speak today. Let me get this going. Everybody see this okay?
We can see you. Yeah, we can definitely
okay. So as Neil said, my lab is focused on factors that regulate skeletal muscle size and function. And there's two primary reasons for this. One is that maintaining muscle function during a variety of pathological conditions, or even just the process of general aging, is going to be important for an individual's ability to maintain functional independence. So this right here is, oh, let me get a laser pointer. There we go. So this gentleman right here is an older gentleman that is climbing a number of stairs, carrying a bag. And without the ability of his muscles to function and generate force, he's going to have problems traveling up these stairs. So in addition to just physical function, preserving the size of the muscle can also be important. So this right here is a survival curve illustrating survival, and I believe this is after cancer prognosis, I believe. And it's showing two individual groups. One group that has preserved their muscle mass, and another group that has lost much of their skeletal muscle. And what you see is that those individuals that have preserved their muscle tend to survive more and for a longer period of time than those individuals that have lost their muscle. So not only is muscle just important for an individual to walk around, carry things, perform activities of daily living, but it also is a strong predictor of how well you're going to survive, particularly when it comes to certain pathological conditions. So muscle size and function can be regulated by a variety of input, both positively and negatively. So positive influences of muscle size and function would include proper nutrients and physical activity, which tend to maintain muscle size and tend to maintain function of those muscles. And then there are a variety of other stimuli that are typically associated with pathological conditions or pathological inducing behaviors that will negatively impact muscle, causing muscle size to decrease and muscle function to ultimately decrease. So when I was a postdoc, I was primarily focusing on these two positive stimuli and how these two stimuli led to changes in muscle size and muscle function. And when I transferred from my postdoc into my own independent lab, I wanted to take on a new area. I had always had a fascination with testosterone and its regulation of muscle because I watched sports growing up, and I could see, particularly with baseball, that testosterone was a powerful, powerful agent that could enhance one's ability to perform. Not only did it make the muscles bigger, but it allowed them to perform better. But, you know, that's not a real societal problem as a whole, I guess I should say. But there's a condition where individuals will lose or stop producing sex hormones to the same level as what is typically considered the normal. And that is a condition called hypogonadism. Now, this can happen in both males and females, but I chose to focus predominantly on the males, and I'll show you why hopefully show you why in a couple slides. So hypogonadism, or the condition where a male does not produce sufficient levels of testosterone, is associated with a variety of conditions. Most notably is aging. And aged males can, up to about 90% of aged males will experience some degree of hypogonadism, whether that's a moderate level or a severe level, particularly when the individual really gets older or gets up in years. Hypogonadism becomes highly prevalent. But there are a number of other conditions as well, such as cancer, where 90% of individuals that are affected by cancer become hypogonadal, cardiovascular disease, obesity, diabetes, and we can even induce hypogonadism pharmacologically. So one of the primary ways to treat prostate cancer is to induce a state of hypogonadism using pharmacological interventions. This is not just this is not limited to the aging population, as many younger individuals in their 30s and 40s are also starting to become hypogonadal because of these conditions such as diabetes and obesity. So the loss of testosterone production, or an impairment, I should say, in testosterone production, is going to affect a large number of males in our society. Now, because hypogonadism is a systemic disease, basically what I mean is that when testosterone production is impaired, it's going to affect all the muscles in the body, because all the muscles in the body are going to be exposed to the testosterone. But when we look at what muscles are going to be of most importance when it comes to hypogonadism, I'm primarily going to be concerned about the limb muscles. And when I say the limb muscles, I'm talking about muscles of the limbs, such as the arms and the legs. And there's two primary reasons why I'm concerned about those muscles. One is that those comprise the greatest percentage of total muscle mass. So the limb muscles are about 63% of all the muscles in the body. And two, those are going to be the primary muscles that allow an individual to remain functional. The muscles of the legs are going to allow an individual to walk up and down stairs. The muscles of the arms are going to allow an individual to carry things and move things around in their daily lives. So I'm primarily concerned with changes in muscle size and muscle function of these limb muscles during a state of hypogonadism. Now, because hypogonadism is prevalent, particularly in an aging population, and because it impairs both muscle size and muscle function, testosterone replacement has become a very popular way to counter those effects in a hypogonadal population. So these are data that were taken from a clinical trial back in the early 2000s from an investigator named Arnie Ferrando, where he took hypogonadal older individuals and he randomized them either into a testosterone replacement group or a placebo group. And I think this was after all these measures are about 6 months post-treatment. And he did a variety of functional and morphological measures. Basically, he looked at how strong the individual was after these treatments and how big their muscles were. So there's a couple things that I want to highlight on this graph, is that when they looked at an indication of how big the muscles were in the leg and in the arm, what he found was that after 6 months of testosterone treatment, those older individuals had an increase in the size of the muscles in those particular limbs. The placebo group, on the other hand, continued to lose lean mass at a pretty significant rate compared to those in the testosterone group. So taking testosterone or giving testosterone to these older males and bringing levels back up to where they would be at that you'd see in a more younger population was capable of restoring some of the muscle in those limbs. But it wasn't just the size of the muscle that was important. When they looked at various functional aspects of those muscles, and here we're going to look at the muscles of the arms, the ability to generate force in the bicep in this motion and the triceps in this motion was increased by taking testosterone, whereas those individuals in the placebo group continued to lose muscle function. Down in the legs, looking at the ability to extend the leg out like you are doing a kick, and flexing the knee backwards, they were able to. I'm sorry, these are both leg extensions, not knee flexion. The ability to kick the leg out showed a significant increase in force-producing capability, meaning the muscles were more functional as well as bigger. So this is very positive data, and this is probably why testosterone replacement has become so popular in our culture. But there is a downside to testosterone replacement therapy. So these data are taken from a clinical trial that actually had to be stopped early because of a variety of adverse effects noted in the testosterone treatment group relative to a placebo group. So in this trial, this was older individuals that were being treated with testosterone or placebo. What you can see is that there are a number of adverse cardiovascular events that occurred in the testosterone group that were not really happening, or I should say there were far fewer happening in the vehicle group. So not everybody can get the benefits of testosterone replacement therapy because of these negative adverse side effects. Now, these are just cardiovascular side effects. There are other side effects as well, including the promotion of established cancer tumor growth. So this extends just beyond, this extends beyond just cardiovascular events in terms of the negative side effects that you can see. So because of this, there have been a number of studies that have tried to figure out alternative ways to increase the size of muscle in individuals that are hypogonadal. And the majority of this work has been focused on taking advantage of what's called the putative androgen signaling pathway. If you were to open up an endocrinology textbook or an anatomy physiology textbook, this is going to be the pathway that will be described for how androgens induce their function on a cell. So essentially what this is an androgen such as testosterone will travel to a cell, it'll go inside the cell, and it'll bind to its receptor inside of the cell. When the androgen has bound to the receptors dimerize and they travel inside the nucleus, where they can change the types of genes that are expressed inside of that cell. So this is the predominant way that people think testosterone functions inside of not only all cells, but including skeletal muscle. So there's been a lot of work to develop what is called a selective androgen receptor modulator. And what this is a chemical that is supposed to selectively bind to and activate androgen receptors in muscle without binding and stimulating the androgen receptor in non-muscle tissue. Now, this has primarily been associated with differentiating activation in the prostate versus muscle. And if this is the pathway by which testosterone makes muscles bigger, this would make sense that this would be a plausible pharmacological way to treat individuals that are hypogonadal. The problem is SARMs have not been shown to work in humans. Now, they will increase overall lean mass in a human, meaning that they'll make lean tissues like the bones and tendons bigger. But when they look at the size of the muscles and the strength of the muscles in the limbs, they don't seem to have an effect on those particular muscle types. And When I started to get into this avenue, I was kind of surprised that many groups and pharmaceutical companies have really gone down this rabbit hole of SARMS and the development of SARMS because of some other data that I had come across regarding how testosterone regulates limb muscle mass. Oops, wrong direction. These are data that are taken from rodents that have a functional androgen receptor in all of their tissues, or rodents that lack the androgen receptor in their muscles. So basically, the receptor for testosterone is deleted from the skeletal muscle only. And much of the work when it comes to how androgens regulate muscle mass is conducted in a muscle called the levator ani. The levator ani is a muscle that sits in the base of the pelvis. And to be honest with you, I'm not really sure exactly what its role is or what it actually regulates. But what you can see is that the size of that muscle is largely dependent upon a functional androgen receptor. So the mass of those muscles in the mice that have a functional androgen receptor is this over here. And the mass of the muscle in those animals that do not have the androgen receptor only in their muscles is about 50% smaller than the muscle in the wild type animals, meaning that even though androgens are present in those animals, the inability to stimulate the receptor in those muscles means that muscle is going to be much smaller. So this muscle right here, because it atrophies or loses size to such a great extent, has primarily been the model system that the overwhelming majority of androgen work has used. But there's a problem. If you look at the size of many muscles of the limbs, there's really no effect when the androgen receptor is deleted. So right here, the quadriceps, these are the muscles of the thigh. And what you can see is there's really no difference in size between the size of the quadriceps in the animals that have the androgen receptor compared to those that do not. The gastrocnemius and the soleus muscle, these two muscles are make up your calf or the back part of your lower limb, of your lower leg, and those muscles are largely unaffected in size by deletion of the androgen receptor. Another group showed similar results in a kind of a different way. They had female mice that have the androgen receptor. They called those the control mice. And then they have androgen receptor knockout mice. And they took these mice and they treated them with either a vehicle or testosterone. And when we look at the animals that have the androgen receptor, these are females that have very low levels of endogenous testosterone anyway, by artificially increasing testosterone levels in those animals, they were capable of increasing the size of the TA muscle. The TA is your shin muscle, the large muscle that sits on the front of your shin. But what they also saw was that in those animals that did not have the androgen receptor, they were still capable of increasing the size of the TA muscle, even though the androgen receptor was not there. So what this means is that it is the presence of androgens, not a functional androgen receptor, that seems to regulate size of the muscles within the limbs, those muscles that are going to be largely important for maintaining physical function in an individual and making up the large majority of total muscle mass. So this is where I got started. I probably, looking back on it, was biting off a little bit more than I could chew, because I was starting with this model right here, where we have hypogonadism causing limb muscle atrophy. And the predominant assumption in the field was that it is a decrease in androgen receptor stimulation that is causing the limb muscle atrophy. But based off of everything I just showed you, that's not the case. So I started with a big question mark in here. And if you look at all the other literature, there has never been a single paper that has defined a factor that regulates limb muscle mass in response to hypogonadism. So when I started, I used a castration model in physically mature male rodents. So in this particular model, you remove the testicles and testosterone production decreases substantially. It doesn't go away because there are other non-testicular tissues that can make androgens, but the large majority of testosterone goes away. And if you were to track the limb muscles in these animals after castration, what you'll see is that you gradually decrease the size of these limb muscles. So this right here is looking at TA mass in the weeks after physical castration of male mice. And what you can see is that compared to the animals that where testicular androgen production was left intact, the mass gradually decreases in size. So the muscles get smaller. So I started much of my work focusing on this muscle right here for a couple different reasons. One, the mass of this muscle was sensitive to changes in testicular androgen production. By removing testosterone, the mass of this muscle decreased. Two, it was an androgen receptor independent muscle, which meant the mass of this muscle was independent of the androgen receptor, but instead relied on the presence of androgens themselves. And the third reason is because this muscle right here was a surface muscle, which meant that I could manipulate this muscle very easily and also measure function of this muscle very easily. So at this point, I got very lucky. I did a couple of small studies to try to get established in this area because I was basically starting from baseline. I had no molecular targets to go after. I had no real understanding of what signals were altered in the muscle when testosterone was taken away. So I was fortunate to get the opportunity to do what's called a large genomic screen in some muscles that I already had. I'm not gonna get into when I say I was lucky. It's a longer backstory that I'm not going to get into, but essentially what I did or what I was capable of doing was taking the RNA from muscles from the TA muscles of mice that were subjected to a sham surgery where testicular androgen production was maintained, and the RNA from the TA muscles of mice that were subjected to castration. And I took that RNA and I did a global screen on these samples to identify which genes are altered in the muscles of castrated mice relative to those muscles from the mice that had testicular androgen production intact. And what you get from this analysis is a list of genes that are that is called differentially expressed. So basically, under my castrated conditions, this is the list of genes that were expressed to different levels than what we saw in the animals that had testicular androgen production. And there's some software that's publicly available called the David Bioinformatics Database, where you can upload your gene list into this software, and it'll take genes that are involved in similar cellular processes and lump them all together so that you get an idea of what functional cell categories are altered under your conditions. And using this analysis, we got a list of what are called functional categories that have a number of genes associated with that, and a number of differentially expressed genes that are associated with that. And this one right here is the one that really caught our eye, biological rhythms. So essentially, in these muscles of castrated mice, the expression of genes that are involved with biological rhythms were differentially expressed. So at this point, we started to go, well, what is a biological rhythm? We have essentially 24-hour cycles in our metabolism within our whole body and within each individual cell of our body. And these changes in metabolism that occur across the day and across the evening are regulated by cellular clocks. When I say a cellular clock, I'm talking about some machinery within each cell that is turning on or promoting the expression of genes that regulate metabolism during the day and regulate the expression of genes that alter metabolism at night. And these cellular clocks respond to cues that are developed by the nervous system, predominantly in the brain. And the brain senses changes in light and darkness to help regulate those peripheral clocks within all of the tissues. Now we're talking about the clock machinery specific in skeletal muscle as being differentially expressed under conditions of hypogonadism. Now I had absolutely no idea what this all really meant when I got into it. I thought it was interesting and had heard a couple seminars at previous conferences related to biological rhythms. And at this point, I had transferred here to FSU, and I happened to be very close proximity to a colleague down at the University of Florida named Karen Esser. Karen Esser is the world leading expert in circadian biology, specifically in skeletal muscle. And I contacted her out of the blue. I told her about my findings. I asked if she'd be willing to meet with me, and she agreed. So I needed a break from Tallahassee, so I drove down to UF and met with her in an afternoon. And she described what it would take to really understand what was happening inside of the muscles related to the muscle clock under my castrated conditions. And about this time, I was fortunate enough to get some funding from the Institute for Successful Longevity here at FSU to conduct these initial pilot experiments. So what we did to look at how the clock was disrupted in the muscle when testosterone levels were reduced, we took physically mature male mice that were about 14 weeks of age, and we subjected them to either a sham surgery where we left testicular androgen production intact, or we castrated them to reduce testosterone levels in circulation. We let these animals recover for 8 weeks, and then we did what I like to call a team building exercise between my grad student and I, where we took tissues from a subset of these mice every 4 hours across an entire day. So we were, that's the team building aspect, because we spent an entire day together collecting tissues every 4 hours from a subset of these mice. So in the next couple slides, what you're going to see is you're going to see a bar that looks like this, where we have half of it in black and half of it in white. Anytime you see the black, it's going to represent a nighttime time point. Anytime you see a white portion, that's going to represent a daytime time point. So the meat of this, and I'm not going to go into the specific details of this particular, of the results of this study, but overall, what we found was that the clock within the muscle, which regulates daily changes in gene expression throughout the day, cycled in a very robust manner in the animals where testicular androgen production was left intact, meaning that the clock was turning on increasing the expression of genes to a large extent, and the expression of those genes were coming back down to a lower level to a much greater degree than what we were seeing in the muscle of castrated mice, where we still saw cycling throughout the day of these of the function of the clock, I should say, but the amplitude or the degree at which these genes cycled was much more compressed. So it wasn't that the clock was completely stopped in the muscle, it was just that it was disrupted in the muscle. So the next step that we took was to really try to understand what was causing this disruption in the muscle clock under castrated conditions. So we started combing through the literature to try to find whether or not there was a factor that regulated the muscle clock and also regulated muscle size. And we came across a protein known as SIRT1. And I'm not going to get into really, I can get into it afterward, but I'm not going to get into really what SIRT1, the role of SIRT1 in muscle. But what we found were two papers showing that the loss of SIRT1 made the clock go from what we saw in intact conditions where testicular androgen production was maintained to a condition that was very similar to what we saw in our castrated mice. Loss of SIRT1 suppressed the clock. And then we also came across another paper showing that loss of SIRT1 enabled the loss of muscle mass. So we now had a target that when SIRT1 levels were decreased, we saw both suppression of the clock and we saw a loss of muscle mass. So that led us to this initial question right here. Was a decrease in SIRT1 causing the limb muscle atrophy that we were observing under conditions of hypogonadism? So the first thing that we did is we measured SIRT1 protein levels in our muscles from this, what we called a circadian experiment, those muscles from those animals that we sacrificed every 4 hours across a single day. And what we saw was that across the entire day, SIRT1 levels in the TA muscle of mice where testicular androgen production was intact had this near perfect circadian expression pattern where it increased in expression throughout the night. It peaked between at the period of night to day, and then it started to decrease again. And that cycling of SIRT1 was completely gone in the animals that were subjected to castration. So this down here is a what's called a Western blot. It illustrates protein levels where the darker the band, the more protein is there. So this right here shows that in animals that were subjected to our sham surgery where testicular androgen production was intact, SIRT1 protein accumulated throughout the day and then started to decrease again. But those animals that were subjected to castration did not have that normal cycling of SIRT1 expression. So we said, okay, if we prevented this decrease in protein expression, what would happen to the limb muscle mass under conditions of hypogonadism? So the first approach that we used to prevent a decrease in SIRT1 expression was viral mediated gene delivery. So how this works is you can you can make it. I bought it. I bought a virus that expressed or that contained the genetic material encoding SIRT1. And what you can do is you can inject this virus into the muscle. The virus will go into the muscle cells. It'll go to the nucleus and it'll put its genetic material into the nucleus, allowing the muscle cell to make more SIRT1 protein. And how we did this is we took mice and we put them into 3 separate groups. We had a group where testicular androgen production was maintained, called our sham group. And then we had 2 castrated groups where we physically removed the testicles from these animals so that testosterone levels would drop. And in one set of animals, we injected the TA muscle, which again is that shin muscle, on one side of the animals with a control virus. So this virus encodes for a protein called green fluorescent protein. It's just a protein that will fluoresce green under a certain at a certain wavelength. And then in a second group, we injected one of the TA muscles with a virus that expressed GFP as well as SIRT1. And what we found when we looked at the mass of the muscle that was expressing there, that was transfected with our virus, and we compared it in size to the contralateral muscle that was not transfected, is that those animals that were transfected with GFP essentially had no difference in muscle size across the animal. Whether we looked at the transfected muscle or the non-transfected muscle, they were essentially the same size. When we looked at the size of the muscle that was expressing SIRT1, we found that those muscles were much larger, or significantly larger, I should say, not much larger, significantly larger than the contralateral muscle that was not transfected. So this was our first indication that a decrease in SIRT1 expression when testicular androgen production is compromised contributes to a decrease in muscle size. Now, this initial approach was very crude. And what I mean by that is that when we inject these viruses into the muscle, not all muscle fibers are going to take up this virus or be infected by this virus. I shouldn't say take it up, infected by this virus. So this right here represents skeletal muscle, and we've cut the muscle in half, and we see groups of muscle fibers here. And when we inject our virus into the muscle, we stick a needle down into the muscle here, and we inject a bolus of virus into that muscle. And the hope is that that virus will go to all of these muscle fibers and infect all of those muscle fibers, telling all of those muscle fibers to express the gene that you want it to express. But that's not, in reality, that's not really how it works. This right here is looking at groups of muscle fibers. And what you can see is that some are glowing green and some are not glowing green. This is typically what happens when you inject a virus into a muscle, is that it will infect some of these muscles. Those that are growing green are the ones that have been infected, whereas some of the other ones around it are not infected by the virus. So we needed a more robust method of increasing SIRT1 in the muscle so that we could actually make some real functional morphological measures in these under our castrated conditions. So fortunately, there was an investigator at UC San Diego who had developed a mouse where you could increase SIRT1 expression only in the skeletal muscle in an inducible manner. So what this mouse had in its DNA was a SIRT1 coding sequence. So this coding sequence, when translated into a protein, would make a SIRT1 protein. Upstream of that SIRT1 coding sequence was a stop codon. And this stop codon prevented what's called a CAGS promoter from turning on this SIRT1 gene. This stop codon was flanked by what are called LOX-P sites. So under conditions where the animal is just is not treated, this SIRT1 coding sequence, which is an artificial coding sequence, it has a normal SIRT1 coding sequence as well, is basically not doing anything. It's just sitting there. In this animal as well, we had a gene encoding what is called a Mer, which is a Cre is a recombinase protein that I'll show you what it does here in a little bit. And the expression of this Mer was driven by a muscle-specific promoter, meaning that even though all cells of the body had this gene in their DNA, because the promoter was muscle-specific, only the muscle was expressing this Mer. So we could take these animals and we can treat them with an agent that would activate this Mer, in this particular case, tamoxifen. So by administering tamoxifen to these animals, the tamoxifen would go to most tissues of the body. But because this Mer was only expressed in the muscle, it wouldn't do anything in all of the other non-muscle tissues. But in the muscle, what it would do is it would interact with the mercury myrrh. It would go over to these LOX-P sites, and it would bring them together, and it would cut them out of the DNA, meaning that this CAGS promoter, which is essentially the engine that drives the expression of this SIRT1 gene, would now become in contact with that SIRT1 coding sequence, meaning that after this tamoxifen treatment, there should be large levels of SIRT1 expressed in the muscles of these animals and not in all of the other tissues of the body. And this is the importance of this, is that the animals are allowed to develop normally with normal levels of SIRT1, and we're capable of turning on this SIRT1 gene only when we want to. And the results of this look something like this, where again, this is a Western blot showing the expression level of, in this particular case, SIRT1. With the darker the band, the more SIRT1 that is there. So when we looked in, this is an example, these are 3 separate limb muscles of the animals that were treated with a vehicle only, basically a control condition, or that were treated with tamoxifen. And what you can see is that those animals that were treated with tamoxifen have much more SIRT1 in their limb muscles compared to the animals that were not treated with tamoxifen. And the specificity of this model is shown over here in that when we looked at SIRT1 levels in a couple of other different tissues of the body, in this particular case, the heart, lung, and liver, what we found is that there's really no difference in SIRT1 levels in the animals that were treated with tamoxifen compared to those animals that were treated with vehicle only, other than maybe a little bit in the heart. So we now had a way that we could really robustly test the role of SIRT1 in regulating the size and function of the muscles under hypogonadal conditions. So to do this, we took those animals and we randomized them into 2 separate groups. One group was treated with vehicle, which is essentially just, I think it was sunflower oil. The other group was treated with the sunflower oil containing tamoxifen. And we gave we injected these substances into these animals for 5 straight days. And then we let them recover for 2 weeks. So basically we could wash out the tamoxifen for those animals and hopefully restore any type of homeostasis that had been perturbed from the tamoxifen treatment. We then did a measure of body composition on those animals to see how much lean mass each one of those animals had, how much fat mass they had. And then we subjected them to either a sham or a castration surgery. So now essentially what we had at this point was a group of animals that were essentially what I'm calling a wild type animal, a normal animal, if you will. We had a sham group and a castrate group for our wild type animals, and we had a sham and castrate group for our SIRT1 animals. So this allowed us to test whether or not SIRT1, the induction of SIRT1 only had an effect on our outcome measures, and whether or not induction of SIRT1 had an effect on our outcome measures under castrated conditions. We let all of these animals recover for 8 weeks. We then did body composition analysis again, and we harvested tissues for analysis. So the first thing that we found is that lean mass, or non-fat mass, or basically bone, cartilage, tendons, muscle, anything that is not fat in the body was partially preserved in our animals that were expressing SIRT1. So here we have our 2 different groups, our wild type animals, which are essentially our animals that do not have the SIRT1 transgene, or that have normal levels of SIRT1, I guess you could say. And then we have our SIRT1 group, where all of these animals were treated with tamoxifen and SIRT1 levels were increased in the muscles of those animals. And we have 2 different groups for each one. We have our sham condition, where testicular androgen production was left intact. And we have a castrated condition here in blue, where testicular androgen production was decreased by removing the testicles. And what you can see is that lean mass was significantly reduced in the wild type animals by castration. We also saw a decrease in lean mass in our SIRT1 animals, except there was an interaction between castration and genotype, meaning that there was a different level of change in this group compared to this group. And when we looked at the change in lean in lean mass, pre-castration to post-castration, what we found is that the animals where SIRT1 levels were normal, they lost about 12% of their lean mass from the start of the study to the end of the study. But our animals that were overexpressing SIRT1 only lost about 5% of their lean mass. So they were capable of preserving some of their lean mass just by increasing SIRT1 expression. This preservation of lean mass didn't seem to be due to changes in the mass of non-muscle tissues. And what I mean by that is when we looked at the mass of the liver of these animals, we saw a decrease in the mass of the liver of these animals, whether we were looking at the wild type animals or whether we were looking at the mice expressing SIRT1. So the increase in SIRT1 in the muscle didn't affect the mass of the liver. We saw an increase in the spleen weight in response to castration, whether we were looking at the wild type animals or whether we were looking at our SIRT1 animals. And we also saw a substantial decrease in the mass of this very androgen sensitive tissue called the seminal vesicle, where essentially when you castrate an animal, the size of this tissue goes down to next to nothing. It's actually really hard to find in these animals. The only place that we saw a partial preservation outside of skeletal muscle was in the mass of the heart. And what we saw was that castration decreased the mass of the heart in our wild type animals, or the animals that were expressing SIRT1 at normal levels. Interestingly, we did see a partial preservation of heart mass in the animals that were expressing SIRT1, but were also subjected to the castration surgery. But this preservation right here is not going to be enough to account for this very large overall preservation of lean muscle tissue. So essentially what this is kind of indicating is that this preservation of lean mass is most likely preservation of muscle tissue. So the next thing we did is we looked at the TA muscle, or that shin muscle, which we found to be partially preserved when we used the virus to overexpress SIRT1. And what we saw was that the mass of the TA was decreased significantly in our wild type animals, or our animals that are expressing normal SIRT1 levels. But that loss in TA mass didn't occur in our SIRT1 animals. So this is showing right here that increasing SIRT1 levels is capable of preserving the size of the TA. Now, muscle is largely composed of proteins. It's the largest protein storage tank in our body. And when you see changes in muscle size, typically largely what you see is that the changes in muscle size are due to changes in the amount of protein that is within that particular tissue. So here, when we looked at how much protein was in those muscles, we see that the amount of protein within that TA muscle was decreased in the wild type animals in response to castration. But the protein content was largely preserved in those animals that were overexpressing SIRT1. So this preservation of mass that we saw over here by overexpressing SIRT1 appeared to be due to preservation of muscle protein, which is important because the proteins are going to be the most functional element of the muscle that allows it to be functional, to generate muscle force, to maintain proper metabolism. So these two experiments have basically put a check mark that SIRT1, or a decrease in SIRT1 expression, links hypogonadism to limb muscle atrophy. And this is the basis of an R01 grant application that recently got scored in a not so favorable way, for my liking anyway, but I will be able to resubmit it in the future. And what we are now looking at, or what we are proposing to do in the future, is to try to understand what is upstream of this decrease in SIRT1. What is linking hypogonadism to this decrease in SIRT1 expression? And what we're also looking at is, okay, so SIRT1 does regulate muscle mass under hypogonadal conditions. How is it doing it? What is downstream of SIRT1 that is promoting this loss of muscle mass? And the last thing that we're doing is we're making, and I'll be starting this next week, we're making functional measures on these muscles in our animals that are overexpressing SIRT1 and subjected to castration, so that we can see if this preservation of muscle size that we get by increasing SIRT1 corresponds with an increase in the functionality of those muscles. So there's a number of individuals that have contributed to this project along the way. The initial work was conducted by Mike Rossetti, who is a past member of my lab. And the current work is being conducted by the 2 PhD students and this master's student that's currently in my lab. And I also have a variety of colleagues that have contributed this in one way or another, both at Florida State, UF, Oklahoma Medical Research Foundation, and a colleague that provided me with this mouse that allowed me to make all these measures, Simon Schenk. And obviously none of this would have been, I wouldn't have been able to do any of this without any of the funding that I received, both from the ISL here at Florida State, which provided the funds for me to make these initial measures that have basically opened the floodgates for me, as well as the startup funds from Florida State and my previous institution, University of Central Florida. So I'd be happy to answer any questions that you might have.
Thanks so much. Yeah, time to open up with questions.
So I see there's some in the chat box here.
One was answered, I think, already. Bob jumped in to ask what the sham was.
So Alice, I hope, do you still have your question as to how I do it?
No.
Okay.
Bob answered that for me. Thank you. I am having a little difficulty wrapping my brain around this, I will admit, as a basic clinician, because, you know, for us, using tamoxifen is quite common. So the notion of using tamoxifen, for example, in males is really counterintuitive.
Yeah.
So I don't know, maybe Lynn has more insight into this with her work with breast cancer patients, but
So
yeah.
Let me just say that we're only using tamoxifen for that short period of time to turn on that gene, and then we stop the tamoxifen treatment. And we have also done, so this was kind of an issue, because one of the questions we had was, does tamoxifen treatment affect our outcome measures? Is this really a SIRT1 thing that we're seeing? We think it is, because we've used 2 different methods to manipulate SIRT1 and saw the same outcome. But somebody might also say, well, what effect does tamoxifen actually have in your system? And we went back and did this control experiment back in the fall, where we took mice that didn't have this SIRT1 transgene, and we treated them with either our vehicle only or tamoxifen, which should not affect SIRT1 levels because they don't have the transgene. And then we castrated them and looked at whether or not we still saw the same amount of change in our muscle mass and lean mass, or whether or not these changes in overall lean mass were due to our tamoxifen treatment. And when the SIRT1 transgene was not there, but the mice were treated with tamoxifen, we saw the same outcome in terms of changes in lean mass and changes in muscle size. So it was the presence of SIRT1 transgene, not tamoxifen treatment, that was affecting our outcome measures. Now, I do agree that tamoxifen is not the perfect approach because it is going to affect other tissues in the body. And one thing that we consistently find is that the length of the bones in our animals that are treated with tamoxifen are actually bigger, but it doesn't change depending upon whether you castrate the animal or not. It's just a tamoxifen treatment effect. So this is kind of like we're limited by technology at this point. There's probably some other agent that has less off-target effects, but at this point, I don't have access to it or know about it. So this is kind of what we're limited with right now.
Thank you.
You're welcome. And Bob, did you have your hand up?
I did, but it was the same concern.
What tamoxifen is doing.
Two birds with one stone. I like it.
Yeah, let me jump in. This is not my area, obviously, but sirtuin's really important in metabolism, generally across lots of different species. And I know a lot of people in the age area got interested in it many years ago as an anti-aging type of intervention.
Yeah.
And so I assume it means that it affects more than just accumulation of muscle protein, but probably repair mechanisms and other types of, I guess, potential intervention routes for mitigating negative impact of aging processes. So can you say a little more about at least what you know about the function of sirtuin?
Yeah, so the primary function that I'm aware of is that it regulates the acetylation of proteins. And in particular, it deacetylates proteins to alter their function, whether that's deacetylating a histone within the chromatin to allow certain genes to be turned on or turned off, or in the case of castration or the loss of testosterone, what we think is happening is that it is altering the acetylation of a very important clock regulator. And by deacetylating this clock regulator, or under normal conditions, what it would do is it would deacetylate this clock regulator, and it would allow the clock to function normally, because it would change the expression of this very important clock regulator. And that under our conditions where you lack SIRT1 expression, you don't have the ability to deacetylate this clock regulator to the same extent, and you get overaccumulation of this the expression of this clock regulator. And we're seeing that the expression of this clock regulator, when left uncontrolled, can actually regulate muscle mass as well. So, that's the mechanism that I'm proposing anyway for how SIRT1 is how it's functioning in our model system. But typically, it's just it's a deacetylase that will modify protein function or regulate gene expression by modifying protein function to some degree. And when it comes to the anti-aging, there has been a ton of work on it. It's actually kind of funny if you follow a little bit on Twitter. It seems to be very controversial as to whether or not it is an anti-aging agent or not. But I haven't quite gotten into the aging literature to know exactly how that has been proposed, other than just it deacetylates histones and regulates gene expression, or helps to dampen gene expression to some degree.